So we're very happy today to have Dr. Valerie McKenzie with us. She is at the University of Colorado Boulder, right here in town, in the what's called eBio, Ecology and Evolutionary Biology Department. She works in sort of across fields that connect parasitology, disease ecology, conservation biology, and herpetology. She has a lot of interest in amphibians. As I rummaged through her website, I also found she has worked on tapeworms in sharks and the gut bacteria of primates and bats, uh, biome on the skin of bats, as well as amphibians. So she ranges wildly in terms of widely, not wildly, widely in terms of the organisms she works on and the relationships. She's particularly interested in the connections of parasites, pathogens, and symbiotic organisms, and how those connect to animals that move around on their own. And she has studied those things in Colorado and other ecosystems, including tropical rainforests. She she's uses those ideas or those relationships, in particular with interest in human-caused change, climate change, and other kinds of changes to the environments that affect what are the relationships of these organisms. How prevalent are they? How do they change disease prevalence or parasite occurrence in animals? And with an eye on conservation, right? So are there things that humans are doing to the environment that change these relationships that put organisms in danger, animals, amphibians, and so on? Uh, and how can we attend to those issues um, in a rapidly changing world? So she's going to talk about some of those projects tonight. She also works as the graduate program director for eBio, has a lab full of students. They do field work, lab work, all kinds of things. So I think you'll find this an interesting talk that connects relationships of animals and their microbiomes. And welcome very much, Valerie. Thanks so much for joining us tonight.
Awesome. Awesome. Thank you so much, Sandra. Thank you for the invitation to come out. It's super nice to meet all of you. And thank you so much to Carol and Yen and Tori for dinner. That was super fun. But it's awesome to get connected with other people who love local wildlife and are paying attention because, yay, we need more of us, right? Okay, so yeah, I thought I'd talk about a couple of different things and convince you why microbes and parasites are pretty fun, pretty interesting. So these are pictures of me when I was growing up. I got super into animals early on and very obsessed with animals, all kinds. And I moved around the country growing up. We were just talking about that a minute ago. And my dad was big into fishing and hunting, so I had a lot of experience tagging along doing those kinds of things, and very much into animals. And that definitely carried over to what I study now, which is a wide range of animals. And this is also why it's really cool to be able to study parasites and diseases, because guess what? Everything has them. And if you study parasites and diseases, you can study all kinds of wildlife. So it's sort of the sneaky way to get to study everything. But as Sandra pointed out, I actually started studying sharks when I was an undergraduate at the University of Connecticut a long time ago. I was really interested in marine biology, kind of like every other kid on the planet. And I was hunting around looking for something to do related to that. And there was a woman who studied tapeworm parasites living in sharks. And I was like, well, it's marine, sounds cool. And I took her parasitology class and I started working in her lab. And then I discovered parasites and I was like, oh, this is cool. I want to do this. So I got to describe some new species of tapeworms that were living in sharks. And from there, I did that as an undergraduate. As a master's student, I got into studying reptiles and amphibians in the tropics and their parasites. And I continued on that kind of stuff during my graduate work. And then I since then have branched out into other types of mammals and birds and things. But amphibians has been a core in my research for a long time. And so I started studying parasites like tapeworms and other types of classic parasites, but I also became interested in the tiny stuff, all the little microscopic fungi and bacteria and other things. And right around the time I moved to Colorado and started doing a postdoc, all of our tools for sequencing to study the microbes had greatly expanded. And turns out I was good friends with someone who was doing that kind of work. And I started learning from a group of friends how to do that kind of microbiome work. Um, and just to put it in perspective and thinking about microbes, if we think about our human bodies, we have about 30 trillion cells that are our human cells in our bodies. But guess what? We've got even more microbes in our bodies. So we've got about 39 trillion microbial cells living in and on us. And these are mostly our gut bacteria. So if you think about it, we're actually only about 43% human. And the rest of us is microbial. And then if you think about the genetic stuff that comes along with that, like the genes of all these microbes that live in us, massive numbers, large order of magnitude more genetic material of microbes than human genes. So we are walking around as habitat for microbes. And a lot of these microbes are good for us. Turns out, and this is known for humans, but these kind of data aren't really known that well for wildlife, but we do know that it's a really similar thing. For a lot of vertebrate animals, yes, we love them and they're so cute, but they are also loaded with microbes. All kinds of bacteria and flagellates and protozoans and fungi and you name it.
How many, if you include the viruses?
Yeah.
You say 43 million, mostly bacteria?
Yeah, those numbers I just gave you were only bacteria. We're not even talking about the viruses. Good point. Then we just get into nutty numbers. Totally. Yeah. Way more. Yes, microbes. Correct. So when we're thinking about all these microbes that live in and on us and in and on wild animals, a lot of them are, many of them are symbiotic partners that might be doing beneficial things for those hosts, and then some of them might be harmful pathogens. And so just to sort of draw those contrasts and provide some examples, and like you can think about it, some of them are fighting for the good force and some of them are in the dark side. But to think about the good side first, a lot of microbes are contributing really essential functions to our health. There are microbes in our gut that are doing vitamin biosynthesis. They are doing neurologic signaling. They're doing pathogen protection. So they're protecting actually from other microbes. They might be helping with host cell proliferation, food digestion, immune functions, all endocrine function, all kind of things. So this is just kind of the short list of big general things that they're doing. But lots of our microbes are helping us thrive as organisms. This is also true for wild animals. They're doing a lot of the same things. But just to zero in on a couple of cool examples, and this one specifically because it involves literally the light side. Bobtail squid, cool marine little organisms. They have an organ on their bodies that contains symbiotic bacteria, Vibrio fischeri. They look like this. They're microscopic. And these bacteria bioluminesce. And so they produce light in this light organ on the bottom of the squid. And that helps the squid in moonlit nights avoid predation, because if they're lighting themselves from underneath, they blend in with the background of the moonlight above. So predators don't see them. So it's a symbiotic partnership between this bacteria and squid that's co-evolved over a long time. Another marine example are these little microbes that live inside of corals that they need to survive. So corals are little animals, right? When we look at coral reefs, they're a giant composite of these pretty small animals, and inside of their tissues, they have these little microbes. They're called zooxanthellae that are photosymbiotic, and they're photosynthesizing, producing food for the coral animal. And in return, the coral animal is providing like a home for them to live in. But these require each other. They're co-evolved together.
bleaching?
That's right. When coral bleaching happens, sometimes in response to temperatures being too high, often the coral can shed their symbiotic microbes and then potentially starve if they don't get them back in time. That's correct. Another couple of examples of symbiotic partners that aid in digestion. Imagine if your diet was wood, or if your diet was only, hay and grass, or if your diet was only blood, right? Those are very specific diets. And how do you get all the nutrition you need out of that diet? The answer is from the symbiotic microbes in your gut that's helping you to thrive on that one specific type of food and derive what you need. So that's the good side of some of our beneficial partners, but then there's the dark side of some of these guys. So these, you're more familiar with, and all of us are way more familiar than we want to be with this virus over here, right, from the whole pandemic. But there's so many of them. This is an example of Staphylococcus aureus, right? Staph, if you get a bacterial staph infection, or this is human malaria, if you get bit by a mosquito and get malaria. So lots of microbes that are causing disease and that are not beneficial. So we're kind of a little bit more familiar with those. But to put it in the context of wildlife, what we're seeing are some emerging diseases in wildlife that are increasing. And what we mean by emerging diseases are diseases that maybe have moved to a new geographic area and are affecting new host species, or they might be becoming more virulent, or they've evolved some kind of way that they've been increasing. But to give a couple of examples, because there is a pattern here, maybe some of you are familiar, being bird lovers, as of the problems with avian malaria, of what's happened to the Hawaiian birds in Hawaii, getting knocked back by a lot of avian malaria. Or bovine tuberculosis, which is a bacterial infection that's affecting herbivores in Africa. Or distemper virus that's affecting large carnivores also in Africa. Like, for example, in 1994, 1/3 of the lions in the Serengeti National Park died due to canine distemper virus, right? That's not a sustainable type of emerging disease outbreak. That's not going to go well. And then another example closer to home would be like the whirling disease pathogen that's affecting native trout in Colorado and other places in North America as well. And something that all of these things have in common are the fact that what's happening is that there's pathogen spillover. So the birds in Hawaii were exposed to avian malaria because people brought non-native birds and non-native mosquitoes into the Hawaiian Islands, which the parasite then spread from those introduced birds to affect the Hawaiian birds. The Hawaiian birds did not have those co-evolved immune defenses to that disease. Same thing with the bovine tuberculosis. Cattle are not native to the continent of Africa, but people brought them in, and with the cattle came bovine tuberculosis that spread over to affect native herbivores. Same thing with distemper. Distemper virus is mostly spread by domestic dogs that have been spreading it to wild carnivores. And unfortunately, introduced fish have been spread all over the globe, but in North America as well. And that is how we got the whirling disease pathogen in North America. And so there's a trend here, right, that you've got invasive species from one area of the globe coming into an area and spreading their pathogens to affect native wildlife. And this is an increasing threat that we're seeing. All these examples I'm showing are well known, but this is also happening continuously. And so these wildlife diseases are increasing in frequency because of our increasingly globalized world and what we're doing in moving animals around the planet. And so this is something I write about and talk about and teach about a fair amount because I really think that we can do a lot better job generally with biosecurity and reducing the spread of pathogens, right? There's no reason we can't do better than we have been doing now that we know better. So how can we fight some of these pathogens that are causing problems for wildlife? Well, some of the tools we have are known tools that we use in human medicine, right? In some cases, there are vaccines that we can produce for wildlife, but that's really expensive, and it really only happens for a very small subset of animals. Then there are things we can do with trying to segregate animals, and there's even sometimes plans to do culling of animals if there are diseases that we see this with the chronic wasting disease. There's been a lot of discussions about how to control that in North America as well. But one of the things I wanted to talk to you about is something I've been doing that's involves microbes that is a bit of a different approach. And we're trying to sort of open new innovative approaches for how we can deal with wildlife diseases, because a lot of the tools we have, it's, we need more tools. When do you never need more tools? So I wanted to talk to you about amphibians, right? So this is a group I've been studying for a long time, right? Amphibians are one of the 5 classes of vertebrates, right? We've got birds, mammals, fish, reptiles, amphibians. Amphibians are pretty speciose, a lot of species. So about how many, there's something like 10, 12,000 species of birds on the planet. I think that's in the right ballpark. There's about 8,000 species of amphibians. So they're also a large group of vertebrate animals, really important to all our habitats. And this is including frogs, toads, salamanders, things like that. They are declining, unfortunately, and they are declining for a lot of the same reasons that a lot of our wildlife is declining: habitat loss, invasive species, all those types of things. But with amphibians, there's also a pathogen culprit that's also responsible for a lot of declines. And this is a fungal pathogen called Batrachochytrium dendrobatidis. That's a big mouthful, so we call it BD for short. So this is a fungal skin pathogen that gets onto amphibians. So it's microscopic. So we're looking here at a picture of skin cells of an amphibian, and these little bright spots are the fungal pathogen growing inside those skin cells. And this pathogen can, in susceptible species, get onto the skin and start spreading, and it'll grow and multiply itself inside of a skin cell, rupture that skin cell, and spread more zoospores to go infect other skin cells. They can also spread them in the water and infect other individuals. So an infection can really build up quickly on an amphibian. And amphibians, because their skin is so important for them, they do a lot of osmoregulation, balancing their water and electrolytes in their bodies across their skin. If you're disrupting their skin layers, that's really critical to them. And so this fungal pathogen can cause a lot of disease and mortality in amphibians. And we first started seeing amphibian declines. We're first starting to get noticed in Australia and in Central America. It was like the 1980s. And it wasn't until the late 1990s that this pathogen was described and recognized as the culprit. So that's relatively recent, right, that we've learned about this. And as it's been studied more and more around the globe, there's strong evidence that this pathogen was likely spread by human activities. So it's that same mechanism that we were just talking about with avian malaria and distemper and those other types of wildlife diseases. So people have been moving amphibians around the globe for different reasons for a long time. And it looks like that activity is what allowed this fungal pathogen to move around the globe and actually looks like hybridize and create a hypervirulent strain. So people move amphibians around the globe for aquaculture as food source, pet trade. And then the first human pregnancy test was developed using amphibians, using actually African claw frogs, these frogs that are native to South Africa. And when that discovery was made in the 1930s, those frogs became extremely in high demand in research locations around the world and were getting exported out all over the world. And there's some indications that that might have been another route by which this pathogen got spread. But the other big one was bullfrogs getting spread through aquaculture. And the magnitude of this pathogen is pretty large. It's not that hyperbolic to say that, as my friend Joe Mendelson was saying here, that it's an extinction event on the equivalent to the dinosaurs, that chytridiomycosis, that's the fungal disease caused by BD, is a panzootic that represents the greatest recorded loss of biodiversity attributable to a disease. So it's thought that about like 40% of the amphibian biodiversity is susceptible to this pathogen and maybe suffering declines due to this pathogen. It's pretty significant. Imagine if COVID was not only causing declines in humans, but was also causing declines in other primates, rats, cats, dogs, whales, pandas, coyotes, across the span of mammals. What if COVID was causing declines across the span of mammals? That's what's happening with amphibians with this fungus. So it's kind of a big deal. It's happening around the globe. It's happening here in Colorado. So this fungus is widely prevalent across the state. In a lot of the species, particularly in the lower elevations, they seem to be tolerating it okay. And we do have invasive bullfrogs, especially in the lower elevations of Colorado, and they are loaded with this fungal pathogen. So it's fun to see all amphibians, but man, when I hear bullfrogs, all I hear is that they're loaded with a fungus. Our Colorado boreal toads are the ones that have been declining most precipitously from this fungal disease. So these toads are high elevation specialists. So the full range of the toads are sort of this, shown in this map here. So you find them pretty widely across Western North America. And then you've got this pocket of them that hits through Colorado, and notice it's up and down through the Rocky Mountains. And so this is a high elevation specialist, and they used to be super abundant all throughout the mountain areas of Colorado. And they've been declining pretty significantly due to the fungal pathogen. It's a great question. So the pathogen is mostly found just on amphibians. There's some studies where we've done that it might also get carried in things like crayfish and other things. It's possible that it can be in the water and then get spread through bird feathers that might visit one pond and then travel to another pond or things like that. But I still don't think we understand all the ways in which it's spreading across the environment, but it's doing so very adeptly. But infected amphibians is one way that it's definitely moving around. And there are some species that are more tolerant that will have the pathogen and be spreading it around to more susceptible species. Let's see. So have any of you seen boreal toads in Colorado before? Yes, they're very recognizable because they have this lovely light stripe down their back. And they're good size. You can have an adult wolf sort of fill up the palm of your hand. They're quite lovely and handsome, in my opinion. The males smell like peanuts, which is strange. So if you ever catch a boreal toad or find one, just smell it. If it smells like peanuts, it's a male. I don't know why that is, but that's what happens. What else? Do you know how long boreal toads live in the wild? What's your guess? 20 years. It's closer to 20 years. Yeah, Carol. So, yeah, they've been tracked in the wild up to 18 years and in captivity more than 20 years. So these are long-lived animals, pretty tough. And they are up there moving around in these mountain habitats. So you probably recognize this picture. I think this is maroon bells. But this is great boreal toad habitat. So when you get up into ponds and small lakes up above 9,000 feet, all the way up as high as you can go, that's where you might find boreal toads. But they are pretty tough. And they love beaver ponds as their breeding areas because the beaver ponds will, create some more still water that will warm up relative to the other types of rushing water down the mountain. And those more still beaver ponds are the areas where they typically do their breeding. And so as soon as the ice thaws out on these mountain lakes and ponds, they can get in there and start breeding and laying long strands of eggs, and they hatch little tadpoles. And then those tadpoles take about 8 to 10 weeks to develop, and they start out really small and grow, they'll be about this big, and they start to metamorphose. And I love this picture because it really captures something that became very relevant for my research that I'll tell you about in a little while. But notice how some of these tadpoles are still very much in the tadpole phase where they're large and blobby, and other ones have started to sprout hind legs and front legs, and their skin has started to change and become bumpy and rough. But this is very typical. When you have a cohort of eggs and tadpoles that hatch, they don't all develop perfectly synchronously. Some of them are going faster and some of them are going slower. It might just be where they're hanging out in places that are slightly warmer or colder, or all kinds of reasons for that variation. But nonetheless, that variation became very important for my research. So I became interested in studying what microbes live in and on these toads. I'd been studying parasites, but as I got into moving into Colorado, I realized that BD was a big problem here for boreal toads, and I really wanted to study that and understand it better. And I started out by studying the bacteria living on the skin of these toads, and we were asking all kinds of questions. And what got me started on that was a paper that I read by a good friend, colleague now, Reed Harris. He published a paper in 2006. So close to 20 years ago now, where he showed that he could isolate certain bacteria from the skin of amphibians, and that when you culture them in petri dishes, sometimes the bacteria would be really good at inhibiting and blocking that fungus. And so what you're seeing here is on the left, this petri dish plate is a lawn, what they call a lawn, like it's growing like a lawn. Across the plate is that BD fungus. So all that's growing here is that BD pathogen fungus on this plate. On the right, what we have is we've got two different strains of bacteria growing on that plate. And this lower one has this ring of clearness around it. And that cloudy stuff is the BD fungal pathogen. And notice it does not like growing next to this strain of bacteria, this Lysobacter. That Lysobacter bacteria is exuding things into that media that fungus does not like at all. And so when Reed published this paper, I instantly became very interested in it. And I wanted to know a lot more about, well, how many types of bacteria can do that? What do we know about them? What amphibians have this? Do the boreal toads have any bacteria like this? All kind of questions like that. So I started studying that with my graduate students. Turns out we found one that is a really strong inhibitor of the BD fungus. So this bacteria, whoops, wrong way. This bacteria is called Xanthinobacterium lividum, or because it's a mouthful, we call it JLiv for short. And this is a bacteria that's commonly found in soil and water. So it can be a free-living bacteria, but it's also found in and on animals often, including the boreal toads. And when you grow it in culture, when it gets up to a certain amount of density numbers in a petri dish, it turns this bright, deep purple color, which is quite lovely. And that purple pigment that it has is called violacine, and that pigment is also really strongly antifungal. So JLiv actually produces 3 or 4 different metabolites or chemicals that are really strong antifungal agents. So bacteria do this all the time. All kinds of microbes. There's microbial warfare going on all the time, right? Like even in our gut, the microbes aren't all just getting along all the time. Some of them are battling each other, and they do that through chemical warfare. So that kind of activity has been going on since way before we were here, that kind of evolution. But we can sort of harness this in a way to create treatments, probiotic treatments for our toads, which is what I've been trying to do. So we studied all kinds of things related to the toads, and including doing a lot of field work in the mountains with the boreal toads to try to understand different aspects of how do the microbes vary across species, how do they differ across those developmental life stages, from when they're little eggs to tadpoles going through metamorphosis. We studied different species across the world, actually, but boreal toads became our home for a while. And once we found that JLiv was a really strong antifungal bacteria that was naturally found on them in their natural habitat, we created probiotic treatments to give a try in the wild. First, we tried it in the lab, because that's better to do that than run out into the wild populations and just start experimenting with microbes. So we did it in the lab first, and we had great success. We put the probiotics on them. Well, we had to try a lot of different ones. Some of them didn't work so well, but the JLiv worked really well in the lab. And then the Colorado Parks and Wildlife, who've been our collaborators on this, said, you know what, some of these populations are really just crashing. Why don't we just go put the probiotics on them in the wild? And I was like, wait, I was going to ask you for permission and you're asking me to do it. This is great. So we collaborate a lot with them. And because we're out there pouring purple bacteria on our toads, we've called this Project Purple Rain, also in honor, of course, of the late great Prince. I kind of think he would have liked this in my imagination. But yes, in Project Purple Rain, we go around pouring purple stuff on toads. So here is our little field setup where you can see the purple bacteria. This one happens to be a little more lavender. And here's an example of me and some of our graduate students pouring it on these little toads. So what we do is we go out and we find the toads during the breeding season, because when there can be thousands of developing tadpoles at one time in a pond. If you go out to try to find adults, you might find 1 or 2 adults in a day, but you're not ever seeing tons of them. So catching those young of the year tadpoles is a way to get a lot of them and treat a lot of them at once. It's also the pathogen they're most vulnerable to the pathogen as they're going through metamorphosis. That can be a critical time. So if we're treating them during that time, that's what we were aiming for there. So these buckets are full of little tadpoles of boreal toads, and we're putting the bath of the probiotic on them that we leave them in overnight. Here's my graduate student, Tim Corpita. He helped build what we called our little toad hotels, where the toads would be staying for a while. So they're staying in their natural environment. We're just confining them in these tubs, watching their development and treating them and taking some samples along the way. So here's me and the group taking some samples. So we use little cotton or sterile swabs to take a little, same kind of thing you would do to swab your own throat for COVID or strep or anything. We swab their skin, and then we can take that sample back to the lab and study which bacteria are there and what the pathogen is doing. And they tolerate the swabs really well. They go right back out into their environment after we swab them. And here's our team of students and Paul from the Colorado Parks and Wildlife in our little mobile field lab that we built. Okay, so here's some results, some data. So what we're looking at here, each dot in these graphs represents the bacterial community on one individual toad. And so the green ones are toads that were in our control treatment, meaning they got a fake probiotic. The purple ones, of course, purple, are the ones that got our purple probiotic treatment. And so before the treatment, all of the toads across the control and the probiotic treated ones look generally the same, meaning the points are all clustered together and mashed together. But a week after treatment, they're starting to separate. And 2 weeks after treatment, now they're looking pretty different. So that's showing, okay, something happened. Something's changing. And when we look at how much of our probiotic, now the y-axis here is the probiotic, how much of it got onto our toads. Before the treatment, they did have a little bit on them just naturally before we treated them, which is also what we saw before. But then once we treat them, then we saw that probiotic a lot of it could get on them. But notice that these points in 1 week later and even 2 weeks later are all spread out. I was so surprised by this. I thought we dunked them into 3 days worth of baths of tons of bacteria. And some of those toads that got treated didn't get any of it on them, right? So there's these points down at the bottom. And it's like, why did these guys not get any on them? Or why was it so variable? So when we went to look at that more carefully, it became really clear that it was about their development. So remember how I said that they develop not all at the same time, that you'll get a mix of metamorphosis going on. Some of them are lagging and staying slow and not metamorphosing. Others of them are getting that rough, bumpy skin and popping out their legs. Amphibian metamorphosis is an incredible phenomenon. Like they are going from being totally aquatic with gills, then their gills change and become lungs. They shed all of their skin, they're growing limbs, and then their skin changes from being a slimy skin to being a rough, multi-layered, more complex skin. So a lot of changes are happening during metamorphosis. And it turns out that really affects the skin microbiome. Shocking. So when we looked closer, luckily we had taken this data, but what we found was that it really mattered what stage of metamorphosis the toads were in, of whether they successfully got the probiotic on them. So it's only these late stages of metamorphosis where the probiotic got really high. So that's why we saw all this variability. And it just goes back to the fact that they're not all metamorphosing at the same rate. And some of them got the treatment at the right time and others didn't. So turns out the probiotics only stick when we treat the really late stage metamorphs. And this was a really important thing to learn, because if we want to do this in a way that can be effective, turns out you have to do this. You have to be really precise on when you're treating them, which is actually hard, unfortunately.
Would it work even later after they're adult, but it's harder to find them then?
It's much harder to find them. You would only get a couple individuals in the wild at a time. I think that you can knock back the infections, but oftentimes adult toads, if they've made it to be an adult, they've got some level of resistance that they've built up. It's these young ones that can get super wiped out by the fungus because they don't really have their full immune system firing yet. They actually don't get all their adaptive immune functions until way after this. Whoa, wrong button, sorry. Way after this stage. So yeah, they take a while to fully develop. Okay, at one of our sites where we were running this field experiment, there happened to be a fungal epidemic going on at the same time. We didn't know that until we looked at our data later. But at the same time, the fungus was present in our field site and affecting that population. And what we found was that, so now we're looking at, on this y-axis is the amount of the pathogen on the boreal toads, and each dot represents a different toad. And here the colors are the control toads, the probiotic treated toads. And then there were other toads in the environment that weren't a part of our experiment, but they were all developing at the same time. So they never were in our treatments, but they were there in the environment. And so what we see is that over the course of the season, so on the x-axis here, we have time, and when we got to go back and recapture these toads, we kept them in their hotels for a short amount of time. Then we put little tag markers on them, the same kind they use for fish, these little, it's kind of like a little fluorescent tattoo you put on their leg. And then we released them all. And then we'd go back and try to recapture the ones we had marked. Oh my gosh, it's so hard to find tiny toads in that huge environment. That's why I wish there was 200 more points here, but these are all the ones we got. Very, very hard to do that. But what you see here is that the pathogen increases over the time of the field season and then it starts to decrease. But notice that the purple points stay lower than the other points. And so over here on the right, it's statistically significant that our probiotic treated toads had lower fungal disease compared to the controls, both types of controls. This is really exciting to me, that our treatment was actually working to reduce the fungal disease load. And if you can help them reduce that fungal disease load and sort of make it through that first season of life, they have a much better chance of beating the fungus and continuing on. So this was particularly exciting for me. So I've spent a lot of time looking for these little guys out in the field. So if you ever find some little metamorphs, it's pretty fun. In the wild, they will sit up in a clumps like this, and you know what? They look perfectly like a little pile of elk poop. So I think they're trying to mimic elk poop as a defense mechanism. And how can you not love an animal that's defense mechanism is mimicking poop? A long time. Oh, well, we started doing the field work, I want to say in like 2008 or 7. And then some of the lab experiments we were doing in 2010, 2011, 2013. And then these field experiments we were doing in like 2016, 17, 18. But it's taken many tries. I'm only showing you the greatest hits. Yeah. So the question kind of becomes, how can we use probiotics in amphibian conservation more broadly? Like, how can we take this and sort of scale it out? Right? Because like doing this kind of thing where we're going out to a site, this is possible where you can identify sites that have turned, and the Colorado Parks and Wildlife scientists, they monitor these toad populations throughout the state because they are a species of concern in the terms of their declines. So they know when sites have kind of turned positive for the disease. And if you know what sites those are, you can go out and isolate the bacteria that's native to that site and do these kinds of treatments on a localized basis. But it takes a ton of work and you have to then treat them at the correct stage of development. So it's very labor intensive, but it is a possible option. The other option is to also think about treating them in captivity before they're getting put out on the landscape. So the Parks and Wildlife does run breeding programs for the boreal toads where they're breeding them in captivity, and then they're going out to try to reintroduce them to sites that they used to be in. And they've had success with that. But one of the challenges is that the fungus is still out there in the landscape, so they can reintroduce it, and then they might get nailed by the fungus. So if you can treat them before you put them out there and just give them that extra protective boost before you're putting them out there, that's the other way that I think that this tool can be effective. We haven't gotten to do that yet. But anyway, those are some thoughts about, microbes and probiotics and how you can use beneficial microbes for wildlife conservation. This kind of approach is being used for a lot of other types of wildlife too, corals, bees, lots of cool stuff.
Did the beneficial microbe impact any other system?
Yeah, so mostly the answer is not really. So, and the other things are, it's really important to use natively sourced microbes, right? And so that's what we're doing. Because we should know by now that going around putting things that aren't native in new places, not a good plan. But no, we've done some work to try to understand what is the effect on sort of the surrounding microbial environment. And we went out and did that kind of experiment and found that the soil microbes seemed to absorb the probiotic and that it was mostly untraceable within like 2 days. Yeah. The natural soil is loaded with microbes. You can't dump enough probiotic on there to sort of perturb it. Well, maybe you could, but not with you'd need airplanes and dump trucks and things we're not using, Oh.
Remember your questions.
Yeah, I know.
We're getting sounds like a lot of interesting questions from the room, and we'd love it if you'd repeat those, Valerie.
Okay, yeah, I will.
Thank you.
All right. Okay. But thinking about captivity for a second, now I'm going to take a left turn. Are you ready? We're leaving toads and we're going totally left turn. Except, wait, we're not leaving toads yet. We're one more toad bit. We studied, in one of our studies, we showed that the toads being reared in captivity look totally different in terms of their microbes from toads in the wild. It's kind of unsurprising, right? So this is a graph showing that similarity of microbes again, where these red ones are the captive juvenile toads and these other ones are wild toads. And look at that chasm of space between them. Like this is some really huge differences. And that's not surprising, right? If you think about the environment that they're living in this very sterile environment here versus in the wild, they're exposed to all of these natural microbes in the soil and in the water. It's no surprise that they're very, very different. I became more interested in this topic more broadly, and I started thinking about how does captivity affect the microbiome of animals more broadly? Because in captivity, their environment is totally different. They're exposed to different types of substrates that have different amounts of bacteria and microbes. Their diet is very different in captivity versus the wild. In captivity, they're sometimes getting veterinary interventions and antibiotics and things that are definitely going to impact their microbiome. And so we did a comparison looking across mammals of looking at the effect of captivity on the gut microbiome in mammals and found huge differences. But it was around that time that I was doing that study that I read an article in the New York Times. And this was in 2018. And I get the newspaper delivered to my house because I'm old school like that, and I like reading it like that. And this article by Rachel Newer was about, it says that python in the pet store, it may have been snatched from the wild. And this article was all about the rampant wildlife trafficking that's happening with reptiles, right? That there's lots of snakes and lizards and turtles and frogs and things that are being snatched from the wild and being sold in pet stores, and that it's this really rampant thing and it's hard to control. And the author of the paper sort of ended by saying, these wild animals are getting pushed into these pet trade chains, and there's no way to tell that they came from the wild. And I was like, well, I could tell you if it came from the wild. And I was like, oh, wait, I could tell you. Wait, okay. So I started following that idea that maybe I could use microbes as a way to think about whether an animal came from the captive or the wild. So some quick acknowledgements for the parrot work. I wanted to acknowledge funding that we received from the United States Fish and Wildlife Service. They had a program called Combating Wildlife Trafficking. I hope that comes back. An EcoSolve grant from Europe that was awarded to my collaborators, Rowan. So Rowan and Stephen from the World Parrot Trust, some of our graduate students and postdocs at the University of Colorado, as well as my collaborators in South Africa. Huge shout out to all of them. And for our toad project, we had funding from the Colorado Parks and Wildlife, the Morris Animal Foundation, and the National Science Foundation for different aspects of that work. A lot of graduate students participated in this work, and undergraduates as well. And it's been super fun to work with them. Okay, well, thank you so much for listening. I hope I didn't go too much over time, and hopefully there's some time for questions. Yeah. Or do you want the microphone? Is that okay?
This is a somewhat unrelated question, but I was wondering, I had heard somewhere that bullfrogs take 2 years to mature.
Oh, yeah.
Is that true?
It is true. Yeah. Yes, a lot of amphibians will develop and undergo metamorphosis all in one season. But bullfrogs are a species that will overwinter as tadpoles and develop in 2, sometimes even 3 years. So they get very large. So that's when if you are ever out and if you go to any golf courses and you see gigantic tadpoles, those are bullfrog tadpoles. They can be like this big. And those would be year-old tadpoles.
Even in captivity, takes 2 years?
I honestly don't remember. Maybe you could speed it up and force it if, artificially, but in the wild, typically it's 2 years. Yeah.
So Anna has a question. Anna, go
Hello.
ahead.
Hello, can you? Oh.
Yeah.
I don't know if, can you hear me?
Yes, we can hear you.
Okay. Yeah, so I was wondering what we can do to help with the boreal toads. I actually had attended an event where we were required to spray like some sort of, I don't know, fungal antiseptic on our shoes for the boreal toads. But I'm not like aware if you can actually buy that or, if there's something more we can do.
Oh, yeah. Great question. Yeah, we had to do that for any time we were coming out of one site and potentially going to any other sites. And even if you're not always had the practice of cleaning off all of our equipment. And yeah, there's like a quaternary ammonia solution that you can use to spray off the bottoms of boots, or if you're using any equipment that touch the water. Yeah, it's important to disinfect those things for sure, so you can avoid spreading the fungal pathogen around. Yes, we used that as well. If you can't find that kind of a solution, using a 10% bleach solution is also a good idea, but you want to be really careful that you're not exposing amphibians to any bleach. So, right, you want to do your disinfecting very far away from the habitat of the amphibian.
Thank you. Great presentation. Appreciate it.
Back to the room.
Can you hear me? Hello? Does this work for dead stuff too, like for ivory or for the skin of cheetahs, can you tell whether, you know, rhino and elephant horn, are there microbes, like dead microbes on them that could tell you whether or not it was a farmed official elephant that was culled or whether it was a wild one?
Yes, that's a great question. People were able to hear that. Can you use this sort of microbe tracking method on sort of animal parts that aren't living, like ivory or skins? So the short answer is we don't know, but my guess would be that it wouldn't be as effective. That gut microbes, that you want to get them fresh and right, so they're containing all of the microbes that are there and they're not degrading. I think other material is not going to be as effective in this way, right? And there are other forensic tools that they use, like isotopes for ivory in particular, because they understand sort of the geographic distribution of different isotopic variation across that. So they can say this ivory came from this place and this ivory came from that place. So for isotopes can sometimes be good at that kind of geographic referencing, but rearing history is a different thing than geographic history, right? And that's where I think microbes are particularly relevant and useful. But I do think live animals, it's better in that context.
Mute button was blocked. Yeah. Let me see. Yeah, I was curious. Awesome presentation. That was really fascinating. So one, I was curious, like when you showed the discrepancy just between captive raised toads versus wild toads in their terms of their like microbial communities, does this cause like any, like, are they less likely to interbreed because of that, or does this cause any like behavioral differences? And then I was also just curious, if would wildlife trackers be able to like circumvent that microbiome test by giving the animals probiotics that they have?
Great questions. Yes, great questions. So for the toad one, right, we see these huge differences in the skin microbiome on toads in captivity versus the wild. And the short answer is we don't know what all the health repercussions of that are. There's definitely got to be some that are the lack of some microbes that's probably affecting the development of the toads in some ways. Like, for example, immune system priming. This is the same in humans, right? Like, this is why nowadays we understand it's better for kids to go out and be in the dirt and wrestle with their pets and things like that and get more exposure to types of microbes as they're developing, because it's helping prime and develop their immune systems. And when they're growing up in environments that are too sterile, like in we that can lead to sort of more autoimmune diseases and allergies and things like that. We don't know this for wild animals and for amphibians or elephants or whatever, but I'm sure that there are effects of a changed microbiome in captivity, but we don't really understand all of them. In rhinoceros, it turns out there is a change in their microbiome, and it's thought to be diet mediated in captivity. That might be one of the reasons why they are so poor at breeding rhinos in captivity, that there might be like probiotic and diet and microbes that can help with their breeding in captivity. So I know that answer went all over, but the short answer is we don't know all the effects. And then your other question about, could traffickers get around it and like artificially make the microbes look better? Yeah, don't tell them that. No, I mean, yeah, there's probably ways that could get circumvented. But yeah, we're still sort of, this is like we figured out that this is a potential tool. Now making the tool useful and how to implement it, that's going to take a lot more research and stuff.
Okay, I'll keep it secret.
Yes, don't tell.
Okay. Folks on the Zoom, are there any more questions from you? Lots of positive comments, Valerie. Lots of people really appreciating the presentation and the clarity of it.
Thank you.
I do have a question myself, which is, I read recently that breeding, the captive breeding program that Parks and Wildlife is doing here in Colorado for the boreal toads is they've now determined that those released toads are breeding themselves.
Yes.
And I'm wondering if there's any way to know if any of the purple rain protection is going with the next generation. Is there any way that's biologically plausible, or would you have to keep
Yeah.
catching toads?
Yeah, thanks, Sandra. That's a great question. So unfortunately, it doesn't look like there's a lot of like if you treat some treated toads that it's not then persisting and going on to that they'll share the probiotic with other toads in the environment. I wish that was the case, but it doesn't. So far we don't have evidence indicating that that's happening. So unfortunately, it would be nice if it would do that, but it really seems like it's kind of a short-term boost in that probiotic on that individual, and then it essentially kind of fades out. But it's providing that sort of critical boost during a time when they're most vulnerable. Yeah. So short answer is I don't think that part is happening. I wish.
Thanks.
Here.
Oh, here, I'll read it to you.
Do you have any idea what stage you have to apply these probiotics to the toads? Is it the tadpole or?
So you could apply it to any stage, but we're finding in our work that it doesn't stick on their skin and become a part of their skin microbiome unless you're treating them right at those end stages of metamorphosis, when they've got the legs and their skin has become that bumpy type of skin. That seems to be the sweet time of when you can treat them and the probiotic will actually integrate on their skin. Otherwise, if you're treating tadpoles that are younger than that, when they have that smooth, slimy skin, it's like we put tons and tons of probiotic on and it just bounces off. And actually, I think that has to do with the skin microbiome of those tadpoles that's very antimicrobial, that it's actually repelling our probiotic. And we're like, wait, no, we're trying to help you. So yeah, that's the answer.
Sandra, anybody else?
I don't see any others here. Zoomers,
We have
last call.
We have somebody here in the room.
One last question. Do you think the treatment of the frog, this particular type of frog, is going to relate to the other amphibians who have the same issue?
Oh, like, can we do this approach for other species? Yes, and there are other labs doing that. Yes. The answer is yes. There have been a bunch of lab studies in different parts of the world. Some trials that it works, some trials that it doesn't work. And it's been a super interesting research area because it's like, well, why does it work here and it doesn't work there? Or which microbes or which hosts are, and I'm always saying, wait, pay attention to the development. We're finding that that's super important. So it works sometimes and it doesn't others. And it's this active research area, but it hasn't been applied in wild populations very much at all. So our boreal toad work is kind of among the first to try that in the wild.
Sandra, anybody else?
I think we're good here. We learned a lot. Thank you.
Yeah, thank you so much. Thank you, everybody. Thanks for your great questions.
Thank you so much, Valerie. This has been wonderful.
Awesome.